Hi everyone, this is Shubhadeep. Welcome to my YouTube channel. In this video, I will discuss about enzyme. That is a part 2 video on my YouTube channel. So let's begin. Okay, in this session, we will read the classification of enzymes and the enzyme regulation. Okay, so what are the types of enzyme? First is the oxidoreductase, then transferase, then hydrolases, lyase, isomerase, and ligase. There are six types or six kinds of the enzymes, and these are the classification of the enzymes. So let's read one by one. Oxidoreductase. Okay, so by the name uh, you can see this definition that they catalyze the oxidation reduction reactions. Okay, so if you see here, this reaction also involved in the or it also catalyzes the oxidation reduction reaction. So it also transfer electron or hydrogen atom from one molecule to another molecule. So that's why it also catalyzes the oxidation reduction reaction. Okay, so what are the examples? So there are examples are dehydrogenase, NADH, NADP, and reductase, oxidases, and peroxidases. These are the examples for oxidoreductase. Okay, move to the next. Transferase. Okay, transferase catalyzes the transfer of a group such as amino, carboxyl, methyl, or phosphate, etc., from one molecule to another. Here you can see X is the somehow it's a group that is attached to A, and by this transferase, it's a type any type of the transferase is a type of enzyme that catalyze this and make the reaction that it all the product is A is totally separate from X, and X add with X is joined together by this bond with B. So what the transferase do? Transferase do the transfer, or it also moving. It do the moving of a functional group from one molecule to another molecule. So it's a function of transferase. Okay. So transferase, there are several kind of transferase like kinases, transaminase. It's also examples for transferase. Okay. I think you understand. Next, mm, hydrolases. It's quite easy. Hydrolases. So by the name, you can understand hydrolase. It's also involved in hydrolysis. That also breakdown or cleavage of CO, CN, and CN. This type of bonding, and you can see how it's added by OH, and somehow what they do, the hydrolysis, let's learn. Hydrolysis involving mainly involving hydrolysis. It's a catabolic function, and so catalyze the cleavage CO, CN, and CC, this type of bonds, and some other bonds with addition of water. So by the addition of water, it breaks this bond. Okay, right, is called hydrolysis. So some of example of this type of enzymes are lipase, protease, pepsin, chymotrypsin, etc. Okay, move lyase. Okay, what is lyase? Lyase, or the main function is splitting chemical into smaller parts without using water. What the difference from the hydrolases and lyase? Hydrolases hydrolases it also do the same kind of work, but here the involvement of water is responsible in the hydrolases, but in lyase there is no involvement of water. So splitting chemical into smaller part without using water, so it's also without it also function, this function is without water. So let's see, here it is, this is a chemical or large chemicals, and it also break these bonds and make such kind of double bond and this type of bonds and make it very smaller part. So splitting chemicals into smaller parts without using water. So catalyze the cleavage of CO, CC and CN same as before by means other than hydrolysis. So here the hydrolysis is not happening or oxidation, giving rise to compound with double bond or catalyze the reverse reaction by addition of group to a double bond. Okay. So examples are decarboxylases, decarboxylase and aldolase. Okay. Next is isomerase. Main function of isomerase is the rearrangement of atoms within a molecule. Rearrangement of atoms within a molecule. So if you see this reaction, ABC is type of substrate, and it's after the rearrangement it makes CAB. So it totally reanimates this same kind of molecule, but just the molecules or atoms are just ends there or take different space for their location. So here just each and individual molecule just change their location or exchange their location. So this is the rearrangement of the atoms within the molecules. So examples for these kinds of the enzymes are phosphohexose isomerase and mutase. You also read this type of enzymes in when you uh, when you have when you read the glycolysis. Then you also read this type of enzymes. Okay, move to the next. Next is ligase. Ligase, another name is synthetase. So ligase means somehow it ligate ligation. So here by the cleavage of ATP or some of hydrolysis of ATP, it just join together two molecules or more molecules, just two molecules coupled or joined by breakdown of ATP or hydrolysis of ATP, or you can say the cleavage of ATP. Uh, so then it joins together, A and B join together and make AB, and then by after hydrolysis ADP and PIR also remains. Okay, next, enzyme regulation. So let's start enzyme regulation. uh, let's know the definition. This is the process by which cells can turn on, turn off, or modulate the activities of various metabolic pathway by regulating the activity of enzyme. It is the enzyme regulation. Okay. So different types of enzyme regulation or methods are also here. The allosteric enzyme regulation, a reversible covalent modification of enzyme, and proteolytic activity of enzyme, feedback regulation and inhibition, and last is the regulation by isozyme or isoenzyme. Okay, let's see the allosteric regulation of enzyme. So what is the allosteric regulation? A type of enzyme regulation by the reversible non-covalent bonding of regulatory molecules. Let's see this picture. Here, this is the enzyme, but it has two subunit. One is catalytic subunit, and another is regulatory subunit. So here you can see the substrate cannot bind with this part because the conformation is not suitable with this configuration. Okay, so after this is the regulatory subunit. After joining the allosteric modulator with this regulatory subunit, then the enzyme becomes active. Then it's become active, or then it also make a signal on a catalytic subunit that also make the changes or makes conformational changes with the shape of the substrate, and then make it the suitable formation or suitable uh, configuration by attaching this substrate, also make a suitable structure as the substrate can attach with it. So This is the function of allosteric. It is the allosteric regulation. You can see that after adding this modulator, if you don't put this, it can't get the shape like this. So it can't add. Then the substrate will not add in this part. So it's very important that the allosteric modulator, or it can be said the allosteric regulator, after binding it, it also changes this catalytic subunit, also change its configuration, and then substrate will join, or it will allow to join this part. Okay, this is the allosteric regulation of the enzyme. Okay, and it's very interesting. Allosteric regulation has some important characters. If this modulator is same as the substrate, then it will be called to homotropic allosteric enzyme. And if this modulator or this allosteric regulator is different from substrate, then it will be called heterotropic allosteric regulator. Okay, sorry, allosteric enzyme, heterotropic allosteric enzyme. So this was the allosteric enzyme, and allosteric enzymes are usually larger and more complex than non-allosteric enzymes. And allosteric enzymes do not follow the Michaelis-Menten equation or Mendel kinetics, and they show a sigmoid curve uh, when the velocity is plotted against the substrate concentration. And the Lineweaver bar plot of an allosteric enzyme will be upward concave shaped. And the feedback inhibition is a type of allosteric regulation. The feedback inhibition is a type of allosteric regulation. Okay. Then another is the reversible covalent modification. What is the reversible? Here the reversible covalent modification. Let's see. It's very interesting. Reversible covalent modification, here the catalytic activity is modulated, also modulated this catalytic activity is modulated by reversible covalent modification. Before you in the allosteric regulation there was non-covalent modification, but here the catalytic activity is modulated by reversible covalent modification of enzyme. And more than 500 different molecules are known to modify enzymes by this methods. Okay, so how the regulation is going on, let's see. Here you can see this is the enzymes, and there are the several types of enzymes also here. And there will be separate enzymes for adding the removing modifying groups of groups to enzymes. So most common modifying groups include the phosphoryl, acetyl, uridyl, methyl, amide, carboxyl, and carboxyl, yes, carboxyl, and uh, hydroxyl sulfate. This type of group also add by adding this type of group, and then makes enzyme active. So let's see, the common is very common is the phosphorylation. The most common type of addition of phosphate to the enzyme specific group, like some of tyrosine, serine, threonine, and histidine residues of the protein. So then the enzyme make active. This after addition, or you can say most common type of addition of phosphate group to tyrosine, serine, and threonine or histidine residues of protein. Then it make active. Then it make active. This is the after phosphorylation the enzyme become active. Then here another this enzyme after adenylation, you can see the enzyme become active. And here the uridylation that makes uridine add with this enzyme. So here there is specific type of group where the uridine will add. So some you can read like if you see the adenylation, in the case of adenylation there will be uh, addition of adenine to tyrosine residues of a protein. If you see the ADP ADP ribosylation ribosylation, then you can see the addition of ADP ribose to arginine, glycine, or cysteine residues. Okay. So there are also addition of this type of group to several group, and methylation, and some ADP ribosylation, uridylation, and there are also this type of uh, modification also called covalent modification. And such kind of molecules that also after adding this phosphate, as before, you can see this enzyme become active, and after adenylation, or this enzyme become active, and after urylylation the enzyme become active, and this enzyme, this ATP ribosylation, so that makes active, so and so on. There are 500 different types of molecules as known, that also modify enzyme this method. Okay. Next is the proteolytic activation of enzyme. It's a very important, the proteolytic activation of enzyme. And here you can see this is the inactive, or this is the uh, inactive form of enzyme. It's also called the proenzyme or zymogen. So in some, there'll be some question on zymogen. What is zymogen or proenzyme? The proenzyme or zymogens are inactive form of enzyme. So you can see there is a cap-like structure on the that also put or also stay in this part and make this enzyme inactive. So the active site is blocked, totally blocked by this polypeptide segment. So after this polypeptide cleavage, you make this enzyme active. After this polypeptide cleavage, you make this enzyme active. So then the substrate will bind this active site. Okay. So it's a zymogen activation by proteolytic cleavage. And some examples are chymotrypsinogen from you can uh, convert it chymotrypsin, trypsinogen to trypsin, and pepsinogen to pepsin. If you want to know about the significance, then I will tell you that it also helped to prevent the autocatalytic damage of cellular components, and it can be converted to the active site when it's needed. So it also it can it's very specific, so it can be converted to active site when it's needed. Okay. Next is the feedback inhibition. Okay. So the feedback inhibition is a type of allosteric regulation. So in some of multi-enzymatic pathway, the regulatory enzyme is specially inhibited by the end product of this pathway, and whenever the concentration of the end product exceeds, whenever the concentration of the end product exceeds the requirement of the cell. So when just take this example as L-threonine, and there are multi-enzymatic in this multi-enzymatic pathway. There are also several enzymes also involved in this process. The first enzyme is this E1 is this enzyme that also makes A, and then E2 makes B, and E3 makes C, and E4 makes D, and E5 makes this L-isoleucine. Okay. And just assume this L isolation is very much in the cell, or its concentration is very much. So what it do after exceeds its concentration, it make a signal, or it also block this fast the at the first this enzyme which makes the A from this threonine, L-threonine, that makes B after not B, that which makes A. This enzyme it blocks this enzyme, that is threonine dehydratase. So it block it first. It block it. So after blocking this, the whole process will halt. So this is the feedback inhibition process in some of multi-enzymatic pathway. That some multi-enzymatic pathway, the regulatory enzyme is specially inhibited by the end product of the pathway whenever the concentration of the end product exceeds the requirement of the cell. So it's a type of allosteric regulation, or I have already said the heteroallosteric regulation, where the end product is also as a act as a uh, modulator, an allosteric modulator. Okay, next move to the feedback regulation. Feedback regulation here, the enzyme is not directly inhibited by the end product. It's very interesting, as it does not directly, it does not directly work on the enzyme directly inhibit it, it does not directly inhibit, or it's not directly inhibit this end product by the end product. End product reduces the concentration of the enzyme, inhibiting the synthesis of new enzyme molecule. So it inhibits the concentration, concerned gene, the concerned gene, which genes produce the enzyme, it just inhibit this gene. And it doesn't make the such kind of enzyme. So in this way it reduces the concentration of enzyme by inhibiting the synthesis of new enzyme molecule. Okay, so it just inhibit, or just which gene just interfere with this enzyme, just block this gene, or just inhibit this gene, this synthesis of this protein from this gene, or synthesis of the enzyme from this gene, after after blocking or after inhibiting. So the it also reduce the new coming enzymes that are also produced from this gene. So this is the feedback regulation. They're also different from feedback inhibition, totally different from feedback inhibition. Okay, next or last is the isozyme. Isozyme is a very important. This enzyme have same catalytic function but have different amino acid sequences, Isozymes are enzymes doing similar catalytic functions but having different amino acid sequences, and they have different kinetic parameters also. If you calculate the Km values, Vmax values, and V0 values of isozymes, that also varies. It also varies. And here, the in mammalian in mammalians, lactate dehydrogenase is a classical example for isozyme, and they are also very much. There are also subunits. There are like A4, A3B, A2B2, A1, B3, and B4. Just see, there is A1, A2B, A2B2, AB2, B4. There are so lactate dehydrogenase has typically, there are also some subunits. So there are also kinetic parameters are also there. These isozymes varies, and some different tissues express different isozymes. Different tissues express different isozymes appropriate to their metabolic needs. When the metabolic which type of isozymes needs, they makes this type such type of uh, for their tissues. So by regulating the relative amount of A and B subunits of cell, so they also regulate the enzyme activity of their specific tissues. Just I give an example. In liver, the A4 type of LDH also predominates, and in the heart, B4 type predominates, and in the brain, A1 B3 type is most common one. Okay, so it was all about isozymes. So here just our session has been ended in this session. So if you get benefit this from this video, please like and share with your friends, and just subscribe. Thanks, goodbye.